Well, welcome everybody. This is a real great pleasure and a great honor today to have Professor Gary Cherr, Director of the Bodega Marine Lab, Professor at UC Davis of Environmental Toxicology. Gary hails originally from Sonoma State, where he did his BA in 1979, and then he did his PhD in zoology at UC Davis in 1984. He also did an NIH postdoc at UC Davis in the School of Medicine until '86, and he's been a professor at UC Davis in the departments of environmental toxicology and nutrition. He's an expert in reproductive physiology, effects of environmental stressors. There's so many pollutants that we talk about in the environment, and we've had a number of exposures this term to hearing about lots of different pollutants. And Gary studies a wide range, including nanoparticles now, oil for a long time, which he'll talk about today, looking at some of the most sensitive stages and life forms in the marine environment, where some of the most major effects can occur, and understanding the mechanisms of those effects and the extents of those effects. He teaches courses in developmental biology of marine organisms and the effects of environmental stress on marine organisms in the context of developmental biology. In his teaching, he did a really important thing for the UC system and beyond. For many years, he led this UC Toxics research and teaching program course in ecotoxicology at the Bodega Marine Lab, and there were many people here in this room that were involved in that course. It was very influential. There's faculty throughout the UC system that were influenced by this, and beyond the UC system. It was a great contribution, and a lot of us learned a lot by having been involved through Gary's leadership. A lot of us also know him through the collaborative work when he was Associate Director of the Pacific Estuarine Ecosystem Indicator Research Program, EPA-funded program several years ago, led out of Bodega Marine Lab. And his role scientifically was to lead the group in biological responses to contaminants. There's a number of us now. I can't possibly name all the connections because there's so many people in this room that have collaborated with Gary and are continuing to do so, and we really appreciate that, Gary. You're a great collaborator. A number of us are enjoying the collaboration and benefiting greatly now with Gary's involvement in the Center for Environmental Implications of Nanotechnology. This is the NSF and EPA-funded center that's led out of UCLA with UCSB, UC Davis, UC Riverside, Columbia, and many other partnering institutions. But what Gary's talking to us about today, beyond all of these other levels of involvement, he's also a world expert in oil spills and oil spill effects on marine organisms. He serves on the Exxon Valdez Oil Spill Trustee Council Science Panel. He recently led the University of California Davis team in collaboration with NOAA, the National Marine Fisheries Service scientists investigating the impacts of the Costco Busan oil spill in the San Francisco Bay. And now he is actively involved as one of the co-leaders in the NCEAS working group on the Gulf spill ecotoxicology issue. And so without further ado, it's a great honor, Gary. Thanks for coming to see all of your friends and colleagues here. And we're looking forward to hearing your talk, Oils and Embryos Do Not Mix, Effects of Petroleum Constituents on Developing Marine Organisms.
Thank you so, oh, that's loud. Thank you so much, Trish. Just better stand farther away here. Actually, that applause at the Bodega Marine Lab, we don't allow applause when a person is first introduced. They only get applause if it's a good enough talk at the end. So if you don't applause at the end, I'll understand. It's fantastic to be here. As usual, I love coming to Santa Barbara. And I don't want to say it's great seeing my old friends, because, but my very good friends. And it's wonderful seeing everybody. So hi, Archer. So anyways, what I'm going to talk to you about today, and for those of you who have not been at the Bodega Marine Lab before or up the North Coast, I invite you to stop by. It is part of the UC system, and it's sitting on a 365-acre natural reserve system, part of the UC Natural Reserve System. And this is a photo of our lab. And in terms of the context of our location and dealing with environmental stressors, we're kind of in a very interesting situation because we're located on the north coast. Let's see, can I turn the lights down a little bit? Or? So we're located on the north coast. Oh, great.
Will the recording still?
That's fine. Okay, thanks. So the Marine Lab's right up here north of Point Reyes. And one of the unique aspects is of the north coast, we're one of 4 regions in the world that has a very active upwelling season to it with high nutrients and very high productivity. But at the same time, we're about 60 miles from San Francisco Bay, which is one of the most impacted, stressed out estuaries in the world. And so a lot of our work, while dealing with kind of the natural environment, a relatively pristine coastline here, we do a lot of work, and particularly my lab does a lot of work within the bay where there's numerous issues. You kind of take your pick of what you want to work on as a toxicologist. But one of the aspects that we have up on the north coast is the movement of water out of the estuary, out into the ocean. And here from the satellite image, you can see sediment coming out of the bay, out into the ocean. And in the wintertime, really starting from about now until upwelling starts in early spring, there's a movement northward of water directly out of the bay. And this is just some drifter. John Largier at the Marine Lab put out some drifters in San Francisco Bay. And within 2 days, they were up off Point Arena, hugging the coastline. And in fact, this is what happened when the Busan spill occurred in November of 2007, that there was some concern. The oil did make it up towards Point Reyes, but it immediately came out and moves northward. So while the Bodega Marine Lab and our area of the coast is fairly pristine, we're also potentially impacted by whatever comes out of the bay. So I just wanted to show that to you. So today's presentation, I'm going to be talking about the response of embryos to exposure are early in development, and then also their responses at later stages of development. And then at the end, I'm going to talk about a case study. This is the Busan spill in San Francisco Bay, where we looked at Pacific herring embryo development, both in the field as well as some mesocosm work at the laboratory, to try to assess what kind of effect that bunker fuel spill had on this indicator species, which is also, as many of you probably know, a very important commercial fish species in the state of California. So just conceptually, I wanted to give you an idea I noticed the colors are wrong on this for some reason, but give you an idea about, because I see these are both yellow and it's supposed to be green, but the idea that, and this is from a review paper that was published by Amro Hamdoun, who's one of the coastal tox trainees. He was in my lab, and Dave Eppel in PNAS. And it basically conceptually shows that during embryo development, there's actually a fairly narrow range in this optimal fitness range where embryos develop normally. And if you start moving either at one end or the other in terms of stress, you'll see teratology. And teratology is just referring to abnormal development. It can be mortality, but it can, typically we define it as birth defects or abnormal development. And so when we look in early development, and this is conceptually what I've adapted this model that they had, that this optimal range is extremely narrow in early development. And if you move much farther either direction of that optimal range, you'll see a dramatic increase in teratology, abnormal development, or mortality of the developing embryo. And later in development, this widens out tremendously. So the optimum is much larger, and you can really see this difference in terms of the later embryo versus this early embryo. So I'll be, just conceptually, keep that in mind, and I'll be showing you examples of that as we move through the talk. So I'm going to be talking about data from 2 different developmental systems that we work with in the lab. One is the sea urchin Actually, we work with both the Strongylocentrodus purpuratus as well as Lidochinus pictus, the white urchin. The Lidochinus work actually started way back with the original MMS, Southern California Education Initiative, looking at long-term effects of oil development in the Santa Barbara Channel. So we've worked with that system for many years. And the echinoderm, as many of you know, is radially symmetric as compared with fish. This is zebrafish, but I'll also be talking about herring today. It's a teleost, and of course they're bilaterally symmetric. And so just to refresh your memory about sea urchins, the sexes are separate. You can see the males up here with white sperm, the females with orange eggs. And during development, first cell division occurs in about 90 minutes after fertilization and proceeds with divisions about every hour, depending on temperature. And this is showing it up to the blastula stage, where it's a hollow ball of cells. And then after that stage, the embryo, the tissues begin to differentiate. So this is a critical transition between blastula and these stages down here where gastrulation occurs. Gastrulation is characterized morphologically by the formation of the gut in the embryo. So it's very easy to look through a microscope and see that. But it's really better characterized in terms of tissue layer development. So it's the endoderm, the mesoderm, the ectoderm are being differentiated, and there's a number of genes that are turned on at this stage. And so this is a kind of a critical stage, and it's thought that gastrulation for all animals is probably the most important part of your life, because if gastrulation goes wrong, you're not going to survive. And then, of course, within about 72 hours, a feeding Pluteus larvae swims through the water. So hatching occurs right around this stage. Zebrafish development, of course, is quite different. It's more similar to the chicken egg, that the early embryo, there's multiple cells sitting up on top of a very yolky egg. So in the sea urchin, it has yolk, but the yolk's distributed very evenly. But in this case, the yolk is so dense that all of these divisions occur right up on top of the embryo. And so you can see this little cap of cells. And then those cells spread out over the yolk, and gastrulation occurs. And then you can see the developing larvae here, eye formation, and so on, until hatching. And in the case of zebrafish, which is a tropical fish, this occurs within 48 hours. So all of these events are happening very quickly. It's more like 5 to 7 days or longer with herring at cooler temperatures. But same basic idea. So what I'm going to be talking about today is the effects of oil constituents, and specifically polycyclic aromatic hydrocarbons, or PAHs, and their effects on development. And PAHs are ubiquitous in the environment. They're naturally occurring from any burning. So after a big forest fire, there'll be major inputs of PAHs into surrounding water. But of course, we think about these. This is the Exxon Valdez. We think about these major releases of oil into the environment. That's certainly a major source. My work in the lab originally started with PAHs looking at creosote pilings in San Francisco Bay. And so these can leach out PAHs for up to 40 to 50 years after being placed in the water. There's chronic inputs. Of course, now we're banning two-stroke motors, but these outboard motors put a lot of PAHs into the water. Again, aerial distribution and deposition, road runoff, and so on. Roofing material and asphalt are a big source. And so these are just, I mean, there's many different PAHs and many different non-PAH compounds in all of these mixtures. But these are some of the more common ones. These 3 to 5 ring PAHs, which are known to be represent quite a bit of the toxicity in water in aqueous systems. And so when we think about pH concentrations in the environment, and this is going from low to high, just as an example. So if you look at seawater offshore, you're talking about part per trillion levels of PAHs. Again, this is primarily through aerial deposition. As you move into, for example, San Francisco Bay sediments, you're in the part per million range. And some other estuaries, start the part per million, a high part per million range in some cases. And then some of the lakes where there's been significant contamination, primarily because of creosote sites or other industrial production sites, can be quite high. And then on the East Coast, and this is the classic example, is the Elizabeth River and New Bedford Harbor, where creosote production sites, you can see you're getting into the part per million, in some cases part per thousand range. And this has been studied fairly intensely on the East Coast and looking at the adaptation of the small fish, the fungulus, the killifish, and its adaptation to pHs at some of these sites. And that's a whole other story, but it's an interesting one. So I want to start off first talking about the response of embryos to oil and oil constituents early in development. And a key point is, these are really just those key stages again, is this early cleavage stage. And it's during early cleavage that the embryonic axis is determined. And what I mean by that is essentially the top or bottom of the embryo is actually determined at that stage. So in the case of the sea urchin, it's radially symmetric. So this is the animal region or the vegetal region. It's the dorsal or ventral region in the fish. And so this is true for essentially for all animal development, is that this axis, which is key to obviously the formation of the body plan and successful development, is determined at these early stages, right up until this blastula stage. And then at the blastula stage, this is where there's the activation of the embryonic genome. So up until this time, during these early cleavage stages, most of the proteins that are being made in the embryo are from existing maternal messenger RNAs. So mom, I kind of describe this oftentimes as mom is packaging lunch for the kids, and they're packed full of this stored messenger RNA that's very stable inside of the oocyte and the egg. And then as development proceeds, they are essentially using the majority of that. There's some transcription, but the majority of that messenger RNA is being translated into protein during early development. Part of this is the rapidity of the cell divisions. And so there's not, the cell cycle is such that you're not transcribing new DNA or transcribing new RNA. You're essentially using what's existing. And then right at the blastula stage is when there's this massive activation of the embryonic genome itself, and then you get new transcription. And although I'm describing this as kind of an all or nothing event, you can actually do this experiment, and we do this with some of our students, the undergraduate students, where you can put in a very potent transcription inhibitor that's essentially a poison in these early stages, and you get a completely normal embryo because they don't need transcription. They're essentially using all of the stored maternal RNA. And then if you keep it in there, they just stop at this blastula stage and they won't go any farther because they cannot transcribe genes that are important for gastrulation. And this is when tissue specification occurs. As I mentioned, the tissue layers are determined, and there's massive differential gene expression. So when I talk about early development in the context of this talk, I'm really focusing in these early stages prior to this differential gene expression. So again, the idea that this blastula stage is right around the time when transcription of the genome occurs. And the classic example, again, we use this in one of our labs in the classroom. It's a really great kind of experiment that if you heat shock, so you do a thermal stress, this is a 1 hour or 30-minute stress of the embryos early in development, 100% mortality. Okay? If you take that same temperature, that same exposure at these later stages, 100% survival. And that's simply because they're able now to transcribe and produce the message for heat shock proteins, which are protective, and they survive. So it's kind of a nice all or nothing experiment that we use in the lab, but it's a clear demonstration. In the case of fish development, this active transcription also occurs in this blastula stage. So this is called the Midblastula Transition. You can see the ball of cells up on the top of the embryo, and it's during this phase that that transcription is activated. And in this case, although you can do that same kind of heat shock experiment, in this case there's also been studies that have looked at P450 genes that are involved in metabolizing organic contaminants. And there's essentially no P450 gene expression prior to these stages. So all through this development, whatever small amounts of P450s are there are coping with the exposure, and it's not until later that they're able to really metabolize a lot of organic contaminants from the environment. So again, this is this kind of idea of stage specificity of responses and stress in the environment. So what I want to do now is talk about the embryonic axis. So as I mentioned, axis determination, and again, defining this as dorsal ventral, the body plan, or animal vegetal, in the case of the radially symmetric sea urchin, occurs during development, and it's a very highly conserved mechanism. So everything from the lowest invertebrate up to humans is basically the same signaling pathway that's involved during development. And it's called the Wnt beta-catenin signaling pathway. So although there can be slight differences, for the most part, this is one of these highly conserved mechanisms that's critical for the animal body plan and is specified early in development. So there's numerous genes expressed, so we're not talking about just a few genes. We're talking about maybe 50 to 100 genes that are turned on during this activation of the pathway. And the key is that it's occurring just in a specific region of the embryo to tell those cells that this is the correct region, whether it's the dorsal region or the ventral region. And basically, it's been known for a long time that if this pathway is perturbed, the embryonic axis will either not be established at all or will be abnormal. And of course, that ultimately is lethal, although the embryos don't just automatically die from that, but they never can develop to a larval stage. They can't initiate feeding and so on. So it's essentially a lethal abnormality. And so this is the basic idea behind beta-catenin signaling the axis. So this is a sea urchin embryo, and these are immunofluorescent images using an anti-beta-catenin antibody, but we'll just focus here on this schematic drawing. And under normal situation, here's the gut, so this is normal gastrulation. There's a select number, and this is in the sea urchin, select number of cells in this region, the vegetal region, that the nuclei accumulate the beta-catenin, and that specifies this vegetal region of the embryo. However, if you induce kind of a global accumulation of this beta-catenin, this protein, which is a transcription factor, to move into the nuclei all throughout the embryo, you get this phenomenon called exogastrulation. So it's kind of like an innie and an outie, the idea that this gut, instead of going inside of the embryo, is actually extending outside. And it's interesting because you can have cultures of these where 90% of the embryos look like this, and they're swimming all around. They're quite bizarre looking, looks like kind of something from outer space. And of course, they can never feed, and they never really go on and develop too much. For those of you that know sea urchin development, there's these internal skeletal rods called spicules. They'll actually form some unusual spicules inside here. But after about 24 to 48 hours, they'll end up dying. And experimentally and also chemically, you can modify things. So instead of having kind of this higher level that's required in some of these other cells, you can restrict it or inhibit it, and then you get something called an animalized embryo. So this is oftentimes called vegetalized because it's overexpression of these vegetal genes here, and so you get exogastrulation, and likewise you can inhibit it and get kind of an animalized embryo. And so for example, selenium, for some reason, will actually induce this animalization, and zinc will do the same thing. So the fish embryo is a little bit, not quite as clear-cut, just morphologically, but the exact same thing happens, that normally this beta-catenin accumulates just in a select number of cells on one side of the embryo, and that determines the dorsal side of the embryo. Okay, so it's just in these select cells right at this one side. And here's a normal embryo. There's the head, and the body's wrapped around. The end of the tail is there. And if you induce massive accumulation of beta-catenin in all of the nuclei, the way that I just showed you in the sea urchin, you get these very bizarre looking embryos. These are called hyperdorsalized embryos. You can see these are the cells here, all piled up at the dorsal side, so that's over-dorsalized, if you will. They're oftentimes elongate. They don't complete gastrulation. You can see here's wrapping around, but the tail never completes, encompassing the yolk. And this is typically what happens when they're exposed to some sort of environmental contaminant or disruption occurs in this range, in the 500 to 1,000 cell stage. Okay, so this is something that experimentally goes way back to the 20s, where people had in developmental biology labs, and this is, as many of you know, marine labs were essentially a place back in the old days of, there was no ecology. It was just simply developing systems to understand basically how cells worked. And so, there's lithium chloride, for example, was something that was found that induced this kind of axis disruption. But the question was, is it known that environmental chemicals can disrupt the axis or not? And this is something we had discovered in the early 2000s when we were working with creosote and these pilings. And so subsequently we went back and did some literature searching, and we found this paper, and I know some of the students out there really need to look at some of these older papers. This is from the Journal of Experimental Zoology from 1948 from the Hopkins Marine Station. And you'll typically see there's one author, and this is, I think, probably about a 48-page paper, essentially, that describes, in detail, kind of walked into the lab today, sat down, got out my bowls, and did my experiments. But it was fascinating because a child had found that he saw that in his cultures of embryos, he kept seeing this exogastrulation, and he was describing it. And at first he thought there was a problem with the water and so on and so forth. Well, it turns out that he came to the realization that there was continuous smoking by members in the laboratory. And so they were just smoking as they were doing their experiments. And he decided to test to find out if the smoking was actually the cause of these abnormal cultures of his embryos. So to test this, they just very, very detailed an experiment where you just basically take 1 or 2 puffs of tobacco smoke. And we're not talking about blowing it on the water. We're talking about blowing it into the very clean, dry bowl. And then, you can even rinse it out and then set up these cultures. And what they found was, of course, they were using lithium chloride back then as a kind of a positive control. And they discovered that tobacco smoke did the same thing. So these are some of their drawings. And you can see this kind of, and this is a number of different conditions which induce exogastrulation. This was tobacco. And he says there in blue that is a point of some interest that something deposited on dry glass from a puff or 2 of tobacco smoke produces the same differential modifications of development as lithium chloride. And lithium chloride being used in the millimolar range. And of course, we don't know what the concentrations, but I guess is tobacco smoke, a puff against dry glass, probably pretty low levels. But there's, as you know, benzoate pyrene. There's a number of PAHs in tobacco smoke. So yes, environmental chemicals, it was published before we did our work with the creosote pilings that PAHs could induce exogastrulation. And so here you can see creosote, the aqueous solution of creosote. This is just a piece of wood taken from an old piling, put into seawater, and then, so it's not a solvent extract, and then that water is filtered, and then it's used to expose embryos, and you see this exogastrulation. Phenanthrene, one of the major PAH constituents, has that same effect. Here's the positive control, lithium chloride, and this is the normal seawater control. And this is just showing that a number of the PAHs have this kind of effect. Creosote is very potent. It's a mixture of many of these. Phenanthrene, fluoranthene, and so on. So it's not just 1 or 2. They'll all have effects, but to varying levels. And when they're mixed together, it's pretty potent. In the case of the zebrafish, and so this is work that Ellie Fairbairn, who's now working, doing some work with nanomaterials, when she came into my lab, she decided she was interested in following up to see if the vertebrate embryo responded in a similar fashion as a sea urchin embryo. And sure enough, actually we kind of predicted this, but just because of the highly conserved mechanism, and if you focus on these two, this is a little hard to see in this slide, but basically earlier in development, this is after gastrulation, but you see this hyperdorsalization, this elongation. Already the cells are starting to degrade the cells of the embryo, so these are going to die pretty quickly. But fluorine, the same thing. You get incomplete gastrulation with the wrapping around of the yolk. And then you also get this phenomenon called bustling or bustled, where it's so dorsalized that all the tissue is just staying up at that dorsal side and it's not really spreading out evenly. So this was classically described by others for lithium, and she described it for PAHs and also a phthalate plasticizer. I'm not going to talk much about that today, but this is the kind of results she received. So it's not, we're not talking about 90%, 80% of the embryos the way we see it in sea urchins, but we're seeing this in 20 to 30% of the embryos that are exposed to these environmental chemicals. So, okay, zebrafish, that's a kind of the white lab rat of the aquatic world, and it's a useful model, but it's freshwater and it's warm freshwater, and our real interests are in marine or estuarine systems. So one of the questions was, what about a temperate marine fish embryo? Do they respond? And so Pacific herring, which San Francisco Bay is the southern extent of their spawning range. And for those of you who haven't consumed this, this is kozonoku, which is the ovary. It's typically brined and is quite popular by the Japanese. It's the main product that the California herring fishery sells, both, well, now in California, but primarily overseas. So it's not the fish themselves. This is how when they spawn, they're very adhesive eggs. This is adhering to algae, which is also a very popular product to consume, is developing embryos on macroalgae. And this is an aerial shot, not in San Francisco Bay because the water is too green to see this, but up in the San Juan Islands. And this is a spawning event, and the white there is all sperm in the water. So these are male herring. These are massive schools, and the males deposit all this sperm, and the females come in then once the sperm's in the water, and they deposit their eggs on substrates. And here's eggs on algae. And some of the substrates also include creosote piling. So this is a piling that's completely coated at low tide with herring embryos, and those are all destined to either die or be completely abnormal. So Ellie found the same thing essentially with herring embryos. It just took a little bit longer because it's cold water, but she saw the same kind of elongation, this hyperdorsalization, and also this bustled kind of an effect. Here's phenanthrene, but she saw this with the phthalates and then the positive control as well. So there really wasn't much difference except the temperature, slower development, and also the chemicals had to be at a little bit higher concentration. So what is the basis of this axis disruption? How are these environmental chemicals, contaminants, affecting the axis? And so this went beta-catenin signaling pathway. So the beta-catenin is a family of these secreted glycoproteins. As I mentioned, they're highly conserved. They have a number of roles, not only in embryonic development, but they're also critical in cardiac development and a number of cancers. And so the beta-catenin has actually been a very important marker for human cancers. Recently, there's a number of human cancers that are beta-catenin positive. So the nice thing for us is there's a lot of tools that are available from the biomedical community that we can use. And one of the most important one is this GSK, or glycogen synthase kinase 3 beta. And it's part of this signaling pathway. It's a serine threonine kinase, and kinases are proteins that phosphorylate other proteins, so they're really important in terms as a regulatory component. And it's primarily regulated by inhibition of its activity, and I'll explain that in just a second. But basically, GSK3 beta can regulate the amount of beta-catenin that enters into the nucleus. Dysfunction or lack of this function has been implicated in not only embryonic development or abnormalities in that, but in development of diabetes, Alzheimer's, and bipolar disorder, as well as cancer. So there's a kind of a broader interest in terms of the biomedical field in terms of understanding this really important kinase and this whole Wnt beta-catenin signaling pathway. So this is showing you kind of the way this works. This is a model showing an unstimulated cell. And interestingly, beta-catenin is always being produced. And as long as it's phosphorylated by GSK3-beta, it is targeted for degradation. So when it's phosphorylated, the proteasome degrades it. It does not accumulate, even though there's some present in the cells at all times, but there's not an accumulation in the cell. And then when the cell is stimulated, in this case by this signaling protein called Wnt, it actually shuts down GSK3-beta. And the details of that are a little bit sketchy in terms of exactly how that works. But once that's inhibited, beta-catenin is no longer phosphorylated, so therefore it's no longer degraded by the proteasome, and it accumulates in the nucleus. It's a co-transcriptional factor, and all these genes are turned on. And so that's what happens in the specific region of the embryo, those cells that accumulate beta-catenin in the nucleus. There's a transcription of either dorsal genes in the vertebrate or these vegetal genes in the sea urchin. And so this is just an example showing this with an anti-beta-catenin antibody. So in sea urchin embryos, you can see just one region, this vegetal region, is labeled with the antibody and the control. And within a very short period of time, exposing them, in this case, this was creosote mixture, you can see that all these nuclei are accumulating beta-catenin, maximum level really even at 4 hours. And you can do these same kind of experiments to try to get a timing on this. And this was of interest because there's also the question of, is there a metabolism of these PAHs that's occurring in the embryo? Or is this the parent compound? And within 2 hours in these cells, you can see the nucleus starts accumulating beta-catenin. By 4 hours, it reaches maximum. So this is a relatively acute, short-term kind of an effect. It's something you can add in these chemicals during these critical points of development and wash them away, and it'll still, these abnormalities will still occur. In the case of the fish, these are confocal micrographs. So this is the zebrafish embryo, and here's lithium, a positive control, and beta-catenin, which is also found bordering cells. You can see that when they're exposed to lithium chloride, the nuclei accumulate. We're showing where the arrows are, and the blue is just a DNA stain, which shows that those are in fact nuclei. So similar kind of response in vertebrate embryo as well as an invertebrate. And again, phenanthrene, a little hard to see the way it's showing up here, but also nuclei are accumulating beta-catenin. So this is very predictive. This marker that's also used across different phyla also works in invertebrates as well as with teleos. Okay, so a little bit about this regulation. And I'm not going to talk about the experiments or any of the experimental detail really, but just to let you know that GSK3-beta, the way it's regulated is it's a kinase, so it phosphorylates beta-catenin, but in turn it's also phosphorylated by other kinases. So when it's actively phosphorylating beta-catenin, it's phosphorylated itself at a tyrosine residue, and then when it's shut down in the stimulated cell, serine 9 is phosphorylated. So this is a kind of a positive-negative type of activity. So depending on what amino acid is phosphorylated, it's either very active or it can be shut down. And so this is just a slide basically summarizing a lot of experiments that many of them that Ellie conducted as well as others in the lab. And basically the bottom line is that inhibitors of GSK3-beta are really potent axis disruptors in sea urchins and in fish. So as one would predict, they work quite well. The sea urchin comes out as one might hypothesize that PAHs induce this downregulation of GSK. So there's actually less of it, and there's an increase in the serine 9 phosphorylation. That phosphorylation is the one that inhibits it. So There's less kinase activity, and there's an increase in beta-catenin. However, in the zebrafish, and this was kind of the surprise, there's actually no change in the amount of GSK3 beta, and serine non-phosphorylation didn't change either. Yet we still see this axis disruption. So there's a kind of a head scratcher there, and we don't really know. We know that there's an increase in beta-catenin, but we're not sure exactly why. Why these are different, we don't know. But there's something different in the signaling pathway. And so we do know that the parent PAHs themselves do not inhibit human recombinant GSK3 beta, so you can take some of this, you can buy some of this enzyme and in the lab do a kinase assay, and there was no effect with the parent PAHs. But as I say down here, what we need to do is basically allow some metabolism to occur, because many of these compounds are very rapidly metabolized by the cells, and see if those metabolites actually have an effect. So there's a lot left to do in terms of figuring that out. So I want to move to the response of embryos to exposure to oil later in development. And I just want to point out here, this is work from Carol Vines in my lab and the National Marine Fisheries guys, the NOAA guys that we collaborated with on the Costco Busan spill. Just kind of a summary, but the idea that specific PAHs cause, and later in development, so this is in this in the time period where the heart is forming, cause these abnormalities, and you can see it right away by the body axis being curved like this. And here you can see this is a herring embryo or larvae. You can see a normal jaw, normal heart, yolk sac is about the right size, and as you increase the concentration of exposure to oil, you can see this curvature develop. You see this edema, or the accumulation of water, around the pericardial region as well as in the yolk. And this is just showing, again, ranging from 0 concentration of oil all the way up to the full amount. And this edema is very apparent. See this large space that develops when they're exposed to PAHs or to crude oil. And so what I'm showing you here is just heartbeats. And so this is a control herring embryo or larvae. And the atrium and the ventricle are here. So this is the yolk sac here. And there's 1-to-1 beating of the atrium and ventricle. And what happens is when they're exposed, you can see there's an increase in space here. When there's cardiac edema, there's a 2-to-1 atrium ventricle block. So the ventricle basically slows down. So instead of 1-to-1, it beats, the atrium beats twice for every 1 beat of the ventricle. So there's a cardiac arrhythmia that's occurring. And this begins to have an effect on circulation of the blood. You get this, start to see this curvature of the spine. And then at high concentrations, you can see there's a complete cessation at times. So the heart actually just stops completely and then starts beating again. And again, you can see this large area that is edemic or has accumulated a lot of water, of fluid. And in that case, again, you get this massive curvature. These will die for sure. These probably will die. They can initiate some feeding, but they don't do well. And certainly later in larval development, before they metamorphose into the adult fish, they're probably not going to make it. So lastly, I want to then, with that in mind, turn to the case study on Pacific herring and the bunker fuel spill that occurred in San Francisco Bay. So for those of you who may not be as familiar with it, this was a situation where the Costco Busan ship spill occurred in 2007. It was early November 2007, about 1 month prior to the herring spawning season. Herring migrate in from the ocean into the bay, starting in late November, early December, and spawn until February and March. Herring spawn intertidally as well as subtidally, right where the oil accumulated. And this was part of a natural resource damage assessment to assess the natural spawn, as well as to try to do some outplanting of embryos in these spill sites as well as reference sites. So before I dive into this, I just want to talk about this, how, and I think it's relevant to the Gulf spill, how it's not until we often have these major disasters, such as the Exxon Valdez spill, that we actually learn something about oil. And part of that has to do with both, I think, the way government and industry work. It's not until there's a disaster and then you have to study it, there's suddenly funds available. Rarely are there funds ahead of time to really kind of understand these kind of situations. And in the case of the Exxon Valdez is really the state of the art of where we are now in terms of understanding oil spills and oil impacts in the environment. And there's no question that the Gulf spill will be a whole new chapter in that. But this is from a paper published by Charles Peterson in 2003 in Science, and it's this idea of these changing paradigms in oil ecotoxicology as a result of the Exxon Valdez spill. And so the idea that the physical shoreline, and that the old paradigm was that that in the marshes that there's fine sediments and it'll be rapidly dispersed and degraded microbially as well as photolytically, that the oil effects are onto fish primarily occur over a very short acute exposure causing narcosis. It's mainly the real low molecular weight PAHs, the 1 and 2 ring PAHs that are really toxic. So as weathering goes on, toxicity goes way down. The oil toxicity to birds and mammals, that the oil effects occur primarily, again, short-term acute exposures, which cause effects on feathers or fur and results in death from hypothermia, which is certainly true, but that these long-term chronic effects are probably nothing to worry about. And in fact, that's not, as we all know now, that's not the case. So that, the physical shoreline habitat, for example, oil degrades at varying rates in the environment. And the example is a paper that was published last year showing that oil in gravel on some of the beaches that were oiled in Prince William Sound is still there essentially in the parent form. So not even microbially degraded to any great extent and barely weathered 20 years later. And so you can dig down and still have this fresh oil coming up. The other relevant to what we're doing, this long-term exposure of fish embryos, and when I say long-term, I mean it's, of course, we're still talking about relatively acute exposure when you talk about development, but the finding that the higher molecular weight, 3 to 5 ring PAHs, rather than these low molecular weight PAHs, at part per billion levels, and when I say that, I'm talking about down to about 1 part per billion or even lower, can have these kind of cardiac abnormality effects, and there's no question that there's population consequences. So you have these, even if it's not mortality of those larvae, certainly their behavior and their ability to escape predation is dramatically impacted, and there's effects on reproduction. And then oil effects on birds and mammals. One of the things that's quite clear is the overall health, along with other environmental stressors. The mixture affects this long-term survival and nesting, but also, especially with these animals, that you have this disruption of the vital social function. So their interactions with each other can be modified tremendously, and that can affect the populations. And for example, just remaining with the nest with eggs. Are they going to stay there or leave? And oil exposure will typically trigger this lack of nesting behavior. And in terms of the coastal communities, not human communities, but this is in terms of ecosystem level. So again, the idea that acute mortality, short-term exposure to oil on the shore, that one can clean that up with the Exxon Valdez, high pressure, hot water, pressure washing of the beaches occurred to kind of get that oil off the beach. And it turns out that those cleanup impacts were much more severe than just leaving it alone. So the beaches that were oiled, that were just essentially left alone, they have, for the most part, recovered, even though there's still this issue with oil that's present underneath. But in the case where they went through and actually pressure washed, those beaches have not recovered. And a lot of that has to do with, in Alaska, in that region, you have this cobble that fits together like a jigsaw puzzle, and that essentially holds the fines underneath. And so when they went through and pressure washed, now there's massive erosion on those beaches, and those beaches are just essentially ruined. So this is just an example of a physical effect. And I think, in hindsight, we can say, wow, what a mistake. Why didn't they figure that out? But again, these are things that in the heat of the moment where you're desperate to try anything, you find out after the fact that that was a major mistake. So in the case of the Cosco Busan, here's a photo of the ship. You can see this tear in the side. This is a situation where the ship, the pilot, headed right for one of the pillars of the Bay Bridge, even though he was being radioed that he's to turn around and change course, and he didn't do that. And he hit that, and it spilled 58,000 gallons of bunker fuel. And you think, well, 58,000 gallons. I mean, that's basically a small swimming pool full. So not a lot relative to 11 million gallons in Prince William Sound or 200 million gallons in the Gulf. But for San Francisco Bay, of course, that's sensitive habitat. That was an issue. Here's Alcatraz Island here. You can see the oil aerial shot around it. And it was deposited in very specific areas, so because the tide went out after the spill. And so Horseshoe Cove here, this is the Golden Gate Bridge, Horseshoe Cove, Sausalito, Richardson Bay is here. So there's oiling along these shorelines. There was oiling on Angel Island, and then also Cove, Kyle Cove up here along, this is the Tiburon Peninsula. And then the reference sites that we used in our studies were around the bend here. This is Romberg Tiburon Center, San Francisco State's lab is up here. And so there were several sites here that we used that still are impacted, obviously. This is the Richmond Bridge, I-580. And so it's not as if there's no PAHs there, but there was no oil up in these regions. And so here's an aerial shot of Kyle Cove. And again, I apologize that the colors are not coming up, but this is all, this bar here is a highly oiled, heavy oiled region, and then no oil here, and then lightly oiled here. And these stations that are shown here is where the natural spawn of a herring occurred about 2.5 months after the spill. And these locations out here are where we placed cages of herring embryos. So we had herring that were brought back to the lab. We spawned them. We fertilized the eggs and put the embryos out into cages. And these were in about a meter of water suspended off the bottom, where these were in very shallow water. Not quite, looks like they would be exposed at low tide, but they were not exposed at low tide. So, but it's certainly intertidal spawning that occurred here. And here's one of these cages. These are aluminum, and there's a sheet, Nitex sheet, that has the embryos, and these cages then to protect from predators are suspended in the water. Algae was collected. Here's divers collecting. And then here's one of our cages suspended out here. And this is hard to see in this slide, but this is macroalgae with embryos attached to it. So this is what was collected and brought back to the lab. And when we looked at the cages, so we thought the cages would be a great model system because we knew the exact history of the adults, and we could regulate everything quite well, and we put these cages at both oiled and non-oiled sites. And sure enough, we saw kind of the classic PAH oil effect of pericardial edema. So if you look at the reference site, and you can basically look at the length of the bars here, which is showing the amount of edema. And you can clearly see that some of these oil sites have a quite large bars, so there's quite a bit of pericardial edema. But nothing as severe as some of those other pictures I showed you. But nevertheless, no question it was there. And so statistically, this is by site, you can see the oil sites, these dark bars here, are statistically different from the white bars. And so the two reference sites here show no pericardial edema from the cages, and the oiled sites all show some, for the most part, show some degree of pericardial edema. So that kind of fit this hypothesis that the sites that were oiled, there would be this kind of effect on embryo development. However, when we started to look at the natural spawn, which really didn't occur until several weeks after we recovered the embryos from the cage, we saw something quite different. So these are the embryos from natural spawn sites. So this is from the reference. And when you bring them in from the field, of course, there's a lot of debris associated with them, but you can see the dark eyes. The embryos are inside of the chorion. This is within maybe 24 hours of hatching. When they hatch out, they look really nice, very transparent, very straight, and really no edema. And this is what we saw in terms of the natural spawn that was brought back from the oiled sites. We saw this premature hatching. These are all obviously dead, and many empty chorions that were left behind, but these are all dead and decaying embryos and larvae. And the few that were surviving had this severe curvature. The arrows are pointing to areas of cellular necrosis, so there's cell death occurring all along the spinal region. There's yolk sac and cardiac edema, and the arrows are also pointing to, you can see this kind of bulbous cell death that's occurring all over the head. And whatever ones were twitching slightly that looked like this, within certainly 12 hours they were gone. So it was quite severe, much different than what we saw in the cages. And the few survivors, again, this is just showing what the reference ones look like. These are from 3 of the oiled sites. Again, this is natural spawn, but this is only less than 10% that actually survived. And you can see these very dramatic malformations. And there's a number of parameters that were looked at here that I'm not going to go into, but it was quite dramatic. So in terms of hatch rates, natural spawn, so the blue bar is the percent normal hatch, and then as a percent of the total hatch with the black bar. And you can see at these 2 oiled sites, there were no normal ones whatsoever, and the ones that did hatch here died pretty quickly. At this one site, there was some percent of normal, and then again, here's the reference. And interestingly, if you look, and this is kind of as part of the NRDA, this Natural Resource Damage Assessment, to try to get at, is this, so one of the big questions is, what was background effect? San Francisco Bay is not a pristine region the way Prince William Sound is, if you want to call that pristine. San Francisco Bay has numerous issues with contaminants and many stressors. So one of the big issues that was brought up by the responsible party was, well, how do you know that this is just not something that's occurring in San Francisco Bay? How do you know it's due to the oil? Yeah, you have this correlation, with the sites that were oiled and non-oiled, but how do you really know? And so one of the ways to get at that is to look over time. So this says 08, but it's the 07-08 spawning season. So there was no normal hatch here at all at these oiled sites. There was that small amount here in 08. And then there's not much we could do because there really wasn't spawning at these sites in 09. But if you look in 2010, they're all back to normal. Okay, so basically this was an effect that was seen right after that oiling period. And then it did recover, which is good news. So the question is, why were the embryos so severely damaged by the Busan spill? In other words, why didn't we just see this kind of classic cardiac edema? Why were we seeing this high mortality rate and this basically some of these embryos just essentially dissolving? So one of the questions we asked was PAH or non-PAH phototoxicity responsible? And what is phototoxicity? Well, it's known that UV light can activate many PAHs once those chemicals get inside of cells and tissues. So it's not necessarily increasing their toxicity out in the water. In fact, UV will tend to break them down. But it's actually once they're inside of cells and then UVA and UVB light hits those cells, it typically is thought to be a result of a singlet oxygen, and you get very acute damage to the membranes. So you essentially get a breakage of the cell membrane, certainly of macromolecules inside of the cell. So it's known in the lab, and there's been a number of studies that have shown this under lab conditions, but nobody's really seen this in the field directly. And so what we did was try to recreate this in 2009, and we set up at the Marine Lab, we set up this mesocosm system where we had a number of these columns that, and this is gravel, and we had also natural gravel taken from San Francisco Bay. We coated it with varying degrees of the Busan bunker oil. And just I kind of wish I didn't have an image to show you, but basically if you looked at this gravel, you would have no clue there was any oil on it. You could handle it. I mean, so we're not talking about gravel that has a sticky, you would never know. that it was oiled whatsoever, even at those low doses. And the idea is that we pumped water, there was a continual flow, we allowed it to weather for 1 to 3 months, and this effluent that was just overflowing the beaker would come down into this tank, and down here we could set down microscope slides with the attached herring embryos. And we had this, what we call like a salad bar cover of plexiglass that was either UV blocking or UV transmitting. So it allowed in about 90% of the UVA, UVB, or blocked out all but about 10% of it. And so we had these set up. And so we could do these experiments in either a UV situation or a non-UV situation. And it sounds simple, but it was really a pain in the neck. But anyways, we won't get into that. But the idea is that if you look, and we used Alaska North Slope crude oil, which is ANS, as kind of a comparison. It's not really a control, but there's been papers published that there is some phototoxicity associated with ANS. And what's listed down here is this is the grams per kilogram of gravel of the oil that was used to oil the gravel. So our highest one, which is still lower than what others have used in the literature. This is the total pHs in the embryonic tissue itself. But you can see that at this 1 gram per kilogram level, ANS oil does have phototoxicity. But at these lower concentrations, there's not really nothing to speak of. However, caustic Busan oil, you can see even at the lowest concentration, there was phototoxicity showing up. And certainly at the middle one, it was pretty significant. Okay, and then at the high concentration, you can see it too, but then everything's being affected. The clean gravel that was used as a negative control, and then also urban gravel that was collected from San Francisco Bay, not near the spill site, no effect in terms of phototoxicity. So there was an effect, and this is what kind of a summary slide of what we saw. So UVT is UV transmitted, UVB is UV blocking, and you can see at these different concentrations these kind of effects that if we block the UV, so the idea of phototoxicity is you can have concentrations low enough that are really not causing any sort of adverse effect, but when there's UV light present, they die. And that was the case with these lower concentrations that we saw with the caustic Busan oil or the high concentration that we saw with the ANS. So again, here's this mass mortality. This is similar to what we saw from the field as well as this. So in this range at these higher concentrations, and we do have some chemistry from the environment. I'm not going to present that today because that's NOAA's data that they're still coming forth with. But the idea is that with UV light, transmitted light, this is the kind of effect you see. Without UV light, yeah, they're abnormal, but you have embryos. Okay, so this is real dramatic. And this more or less explained what we saw from the field. So the summary of this is that, yes, we saw the classic pericardial edema at the oiled sites, particularly when we use these cages. Was it due to the caustic Busan pHs? These are low levels of pHs in the tissues that we measured compared to reference sites, but they're significantly elevated, but they're still extremely low. The body axis defects, this curvature of the spine, and this necrosis, this overall, like I defined it as the solubilization of the embryos, natural spawn was much more severe than what we would have expected to see in any typical pH exposure, even at high levels. And it did come back to normal, though, after that spill year. So that's the good news for the environment, but it's also, to me, implicates the spill in that kind of an effect. The potential involvement of non-pH compounds in bunker oil is a real possibility. So all of the focus on the chemistry was on PAHs because they are the classic marker compounds in oil. However, it is known that non-pH compounds in bunker fuel, and this was published by Jerry Neff's group back in the 80s, where they saw significant acute toxicity from non-pH components of bunker fuel. And that's different from the typical North Slope crude. So it's very possible and plausible that not only toxicity, but phototoxicity could have been due to some of these other compounds. So the photoactivation of the bunker constituents in the environment, in particular in the tissues, is likely, and that's what we're putting forth in terms of this confirmation with these mesocosm experiments. But it also explains the difference between the caged embryos. So these caged embryos had the classic cardiac edema, but they weren't showing any of that just dramatic, again, the solubilization of embryos. Part of it was they were at depth, but the other part of it is they were in a cage. They were even if there was sun penetration, which frankly in San Francisco Bay there's not a lot at 1 meter. But even if they were more shallow, in retrospect, if we knew now, it would have been nice to have put them closer into the intertidal. They were essentially protected from UV light. And so that pretty much explains probably why we saw that difference. So in conclusion, I want to wrap this up. So basically the bottom line is that embryos can show differential susceptibility to contaminants in specific stages. So it's our contention that things like PAHs, and we've also found with phthalates, will cause axis disruption in embryos. And it occurs in invertebrates and it occurs in fish. And it also has, to me, there's a lot of concern in terms of human development. These are the kind of effects in early development where there would be a miscarriage. So for example, people that are heavy smokers, they may have trouble getting pregnant, it could be because if axis is disrupted in the human embryo, there won't be implantation, and actually it could be undetected miscarriages. So it can be diagnostic of specific chemical contamination, although it's probably broader than just PAHs. But we know that it occurs across phyla because of that signaling pathway. And that, anytime you're dealing with this kind of effect that is highly conserved, that you're disrupting a pathway that's highly conserved, there has to be concern everything from ecosystem health to human health. And the same can be said probably regarding cardiac development as well, because what we don't know is whether these kind of PAHs can affect cardiac development in humans and in mammals. And it's something that, well, that's not something I work on. I'd like to see some of my colleagues take that on. Again, it's thought to be that the cardiac effects, that cardiac edema is pH specific, although I wouldn't be surprised if we see other compounds causing that same effect. So the assessment of impacts of the bunker oil spill shows that there was a much greater effect than we would have predicted just simply from the pH contamination, both the chemistry from the water, but also from those caged experiments. So those were kind of the classic effect we expected to see, was showing this cardiac edema, but not showing any mass mortality, especially at this level where we're talking 58,000 gallons of oil that was spread out over a large area. However, now that we know more about this phototoxicity effect, and also sites where these low levels were, the way oil spill assessments are done is people walk the beach and look for oil. And if they don't see it, the beach wasn't oiled. And so, as far as we've come technologically, that's still the standard for much of the natural resource damage assessment, which is, to me, is amazing because, again, I kind of wish I should have brought a bag of each of these gravel, and if I put them out there, you wouldn't know the difference. I guarantee it. And so yet you can still have, at these really low levels, these part per billion levels, you can have these effects, certainly on fish embryos, and maybe having effects also on other organisms living, especially in close proximity with the gravel. So just because a site has no visible oil remaining, it doesn't necessarily mean that there should be no remediation. Now, I'm not saying we should come in and dredge all that gravel out or anything like that, but it's just something that I think we have to move beyond. I didn't see any oil, therefore that beach was not oiled. And lastly, I think as many in the ecotox field know, that embryos are highly sensitive and they're excellent indicators, very low contamination, and particularly of PAHs in the environment. So I just want to real quickly point out the people that did much of this work. So Morelli Pillai, who did the work with the sea urchin embryo and axis disruption. Carol Vines has been in my lab. She's the one who worked on herring originally with the creosote pilings. Professor Athula Rikamanayaka from University of Miami, somebody who works in the basic area of development and the signaling pathway and axis development. And then Ellie Fairbairn from the lab who worked with both herring and zebrafish. And then, of course, the folks from the NIMS lab and Northwest Fisheries Science Center, John and Cardona and Nat Schultz. And I want to thank you for your attention.
What is the transport of the bunker oil spill? It moved throughout most of the bay, had impacts in other places. Did it move up the coast?
Did move up the coast.
Potentially had impacts there.
Yeah, so the question is where did the bunker fuel go? So it did come out of the gate and went north and south, primarily north that time of year, went up to Drake's Bay, and that's pretty much where it stopped. So the high frequency radar, CODAR, was tracking where that was going to end up, and frankly I was pretty nervous. I mean, we were getting prepared to shut down our seawater system if it made it up, but as I showed you at the beginning, you can actually, that time of year, you can have this really rapid movement hugging the shore right up the coast. Luckily, because the bunker fuel is this really nasty, heavy fuel that's almost like a mousse, and it starts settling out pretty quickly, I think that limited its dispersion. If it had been a light, sweet, crude, say like in the Gulf, yeah, we would have seen it, outside the gate, and it would have been moving back and forth with tidal flow. It did oil the East Bay, and that was strictly because of tidal movement. So initially it went out the gate, hit the Sausalito area. As the tide came back in, then it hit the East Bay. But again, it sunk very quickly, and there were a lot of tar balls that were collected that were kind of rolling along the bottom.
It seems like it's great forensic work to understand the impacts of oil. What are the other emerging contaminants or other critical contaminants that these tools can be used to detect the impacts of?
Yeah, so, well, the phthalate data, didn't really want to talk too much about it, but as people probably know in terms of, as emerging contaminants go, phthalates, which are basically plasticizers, make plastic soft, are just everywhere in the environment. If you do environmental chemistry on anywhere, you basically have to just kind of ignore the phthalates because they're swamping out everything, and people don't focus on that. But recent evidence is pretty clear cut that they're endocrine disruptors, and our data showing that phthalate and others are developmental toxic compounds as well. But I mean, there's so that's the other issue that you have in a place like San Francisco Bay. You have metals, right? There's metal contamination. There's endocrine disruptors, there's emerging contaminants, pharmaceuticals. All of those can impact these early life stages. Ah, okay. Okay.